Hello there. This is going to be a short video on the AS1 topic, molecules, an essential topic at all levels in biology. Sometimes your teacher would call this topic biochemistry, so it's the chemistry essentially of biological molecules. And to get most benefit from this video, you'll need access to a set of my notes and a question booklet, and you'll also have the answers as well. I will be making a constant reference to areas in the notes, and also be looking at questions. Now, as I go along, this video is going to focus on proteins. I've looked at the carbohydrates and lipids in a previous video. So this video is going to focus on proteins. Proteins probably being the most important of the different biological molecules that you need to know about. So proteins. Just looking first of all, just on the front page of the booklet here, I have a table which looks at the elements present in the different biological molecules, and that's relevant to the extent that it points out that proteins, as well as containing carbon, hydrogen, and oxygen, which all of the biological molecules, the carbohydrates, lipids, proteins, and nucleic acids, they all contain carbon, hydrogen, and oxygen. But notice that proteins also contain nitrogen, and many of them contain sulfur as well. And that might come up in questions, and certainly you need to be aware of it at A2 as well. Just a possible, looking at a possible question there, at question 1 in my question booklet there, at the very bottom, it asks, suggest a possible role of the R groups shown in the amino acids. Well, just to look at that wee question first of all, the R group is part of the amino acid, and that's often where the sulfur in the amino acid is contained. We'll come back to look at that question again, but notice that proteins contain carbon, hydrogen, oxygen, nitrogen, very notably nitrogen, and also possibly sulfur. So now going directly to proteins and looking at protein structure. So I'm turning to page 14 in the notebook up there. Sorry, page 12 in the notebook. I'm looking at proteins, and I've indicated here to myself just that the questions that are relevant to proteins from the question booklet here are questions 1, 4, 5, 6, 8, 10, and 13. That's questions 1, 4, 5, 6, 8, 10, and 13, and you might indicate that in your notes there at the top of that page so that you can make reference to those questions as you go through the notes. So I've been making reference to each of those questions as I go along at different points. So proteins are macromolecules. That's a term that used to be asked about. It might and might not be asked about in the future. Macromolecule simply means a big molecule. Big molecules, big biological molecules that contain carbon, hydrogen, and oxygen and other elements. And as with the other large molecules in biology, these macromolecules are made up of smaller subunits or monomers. Subunits or monomers. So just in the same way as carbohydrates are made up of glucoses and lipids are made up of fatty acids and glycerol, proteins are made up of smaller molecules, monomers called amino acids. Amino acids. Amino acids are very important molecules. So where do we find proteins in nature, in humans, in animals, in plants, and so on? Well, proteins we associate with muscles, but proteins are present in every cell. As you may have already found out with the structure of the cell membrane, every cell has a cell membrane, and the cell membrane contains phospholipids, but it also contains proteins. So there's protein in every cell in the human body, in the cell membrane of every cell. It's just that we associate proteins with muscles because muscle cells have lots of extra protein. They have extra specialized proteins that can contract and relax to make muscles work. But essentially every cell contains protein, and it's important to know that. It's also important to know that making proteins, and proteins are so important for every cell. It's a big part of the function of every cell to make proteins. Protein synthesis, if we refer to it, and you come across protein synthesis then possibly in the cell topic, where you discussed how the cell made its proteins, and you discovered that the DNA inside the nucleus coded for the proteins, coded for the structure of the protein, and that the proteins were made outside the nucleus, made at a place called the RER, rough endoplasmic reticulum, at units called ribosomes. And cells are always making lots of proteins. Bear that, keep that in mind. Cells are making lots of proteins. So what kinds of proteins are we talking about? Well, the cell is making proteins for its cell membrane. So cells are making proteins to complete its cell membrane. And when cells divide to make new cells, then they need to make more proteins for the new cell membranes of the new cells. So cells are making lots of proteins for the cell membrane. But also very important to remember is that enzymes are proteins. Enzymes are large proteins, and cells are making lots of enzymes. A very large production of enzymes. You might remember from GCSE that in the cytoplasm you had chemical reactions. That was the role of the cytoplasm, chemical reactions. Well, all those chemical reactions are governed or controlled by enzymes. So the cell is making possibly thousands of different enzymes, and those enzymes are all proteins. Other things that you would come across that are proteins made by specialist cells would be hemoglobin. Hemoglobin is such an important protein. It's the protein found inside red blood cells, and it carries oxygen around the body. So hemoglobin, another very important protein. And collagen is a very important protein too, found in ligaments and tendons around the body, and it's a protein with great tensile strength, protein that can withstand a lot of stretching. So proteins then are the subunit of proteins is an amino acid. So we need to know the structure of amino acid. And if of all the molecules in AS Module 1 biochemistry, I would say it's very useful to remember the structure of an amino acid. So amino acid has this structure: R to C to H, so that's a carbon, R to C to H, to C double bond O, single bond OH, and then you've got, very importantly, you've got a nitrogen here, and nitrogen connected to two hydrogens, like that. So there we are, R to C to H to nitrogen with two hydrogens and COOH. So that's useful to learn that structure and know that structure, and it points you very directly to the presence of the nitrogen in proteins. Nitrogen is also present in DNA, but very notably in proteins. So in GCSE, you would have learned things like plants need to take up nitrates from the soil. What do they need nitrates for? Because nitrates contain nitrogen, and that nitrogen then could be used to allow the plant cells to make their proteins, to make their amino acids, and to therefore make their proteins. So proteins contain nitrogen. So now we're going to look at how proteins are made. Basically, at GCSE, you would have said you join amino acids together to make the protein. First thing you need to get here is that there are 20 different types of amino acids. So this is the normal structure, the standard structure for an amino acid, but there are 20 different types of amino acid. And you might have heard of some of them before. They've got names like alanine and cysteine and leucine and isoleucine and histamine and proline and serine and so on. Their names tend to end in E, and there's 20 different types of them. And you might say, well, if that's the structure of an amino acid, then how do you get 20 different types of these amino acids? And the answer is, this grouping here called the R grouping is different in each amino acid. And if you, that R grouping, let's say that R group consists just of one hydrogen. If it consists of just a hydrogen, then an amino acid that amino acid then is called glycine. So there's just one hydrogen here, it's a glycine. For another amino acid, instead of having a hydrogen, it could have CH3 there. So the R group could be CH3, in which case the amino acid is called alanine. And so all you do to get different amino acids is to change this grouping up here called the R group. Change it, and lots of different forms, and that gives you your 20 different amino acids. So in each of the 20 different amino acids, you simply just have a different R group. And question, just looking at a question and written that if you look at question 1 in the booklet there, 1 part B, it says suggest a possible role of the R groups. Well, you could say the R group gives the amino acid its identity. It establishes which amino acid it is. So if you have different R groups, then you have different amino acids. So it gives the amino acid its identity. The other thing you might say is that the R group, notice that we said at the start that amino acids or proteins would have a sulfur. Many of them have sulfur present. Well, there's no sign of sulfur there, but in some cases you have a sulfur in the R group. So amino acids that contain sulfur have a sulfur in the R group. And one that comes to mind is an amino acid called cysteine, cysteine, and cysteine contains a sulfur, and the sulfur is present in the R group. And if you were asked what's the role of that sulfur, well, that sulfur can be involved in something called a disulfide bond. Allows that amino acid to bond to another amino acid by a disulfide bond. We'll look at that again in a wee moment or two. So 20 different amino acids, you'll see them listed there. And I've said at the side, just as I said, you don't need to learn them, and neither you do. You don't need to know the names of these amino acids. Just know that there's 20 different amino acids. Now, if there's 20 different amino acids then, and proteins are made by joining amino acids together, then you can understand that if you have different sequences of amino acids joined together, then you might have to that might be how you get your different proteins. And the fact that you have 20 different amino acids gives you sort of endless opportunity for making slightly different proteins simply by varying the sequence of the amino acids in the protein. Now, to understand that how you go about constructing your amino acids into a protein, we're going to look first of all just at joining 2 amino acids together. If you join 2 amino acids together, then you get something called a dipeptide. And I'm not going to draw that on the board, it's take too much time simply to draw it. It's there in the notes on page 13. And as I say, you need a set of these notes to be able to make use of this video, really. And you'll see there, it said 2 amino acids can join together in a condensation reaction to make a dipeptide. So there's an unusual word. Dipeptide suddenly appears in the discussion. So one amino acid joined to another amino acid. It involves a condensation reaction where you remove a molecule of water between an NH2 group of one amino acid and a COOH group from the other amino acid, and you then have the 2 amino acids joined together. The bond that's formed between the 2 amino acids is called a peptide bond. So the 2 amino acids joined together is called a dipeptide, and the bond between them is a peptide bond. And that's a condensation reaction, so it involves the removal of a molecule of water. That's a dipeptide. If you join another amino acid to those 2 amino acids, then you get a tripeptide, 3 amino acids joined together. And if you keep joining amino acids, then you get a polypeptide. So polypeptide, then a polypeptide is just a long chain of amino acids, all joined one to the other. Many amino acids joined together. Now, as I said a moment or two ago, there are 20 different amino acids possible, 20 different possible amino acids. So you can understand that this, if you're making a polypeptide by joining amino acids together, you have a very large choice of amino acids, and of course you can use amino acids twice. You can have them in the sequence wherever you want, and with 20 different amino acids in, you've got a very large number of different sequences of amino acids when you join them together. A polypeptide, so when you create a polypeptide, you join amino acids together. It may be a short polypeptide. It may just have 20 amino acids joined together, or it may have 50 amino acids, or it may have hundreds of amino acids. And many proteins contain polypeptides that have hundreds of amino acids long. So note at this stage we're really just talking about joining amino acids together to get a polypeptide. We're not calling what you form in the first place a protein like you would have done at GCSE. It's a bit more complicated than that. So you're really just making a polypeptide at this stage, joining amino acids together. And you'll note there in the notes I've said, each polypeptide has a very precise sequence then of amino acids in its structure. So if you were making a polypeptide, joining amino acids together, and I asked you, what is the primary structure of your polypeptide? And you would say, that's the sequence of amino acids in my polypeptide, the precise sequence of amino acids. And if I said, tell me about the primary structure of your polypeptide, I basically would be asking you, what is the sequence of amino acids in your polypeptide? So the term primary structure relates to a polypeptide, and it's the precise sequence of amino acids in that polypeptide. And it turns out that eventually when we get our protein made, that it's that precise sequence in the polypeptide that's all important in determining the type of protein that you get produced at the end. So that sequence of amino acids in a polypeptide essentially determines the protein that you get produced eventually. And the other thing that comes across maybe in A2, you'll come across in A2, is the idea that you could have mistakes in the sequence. And you would have a mistake in the sequence. A cell could make a polypeptide that's got the wrong sequence of amino acids. And that would be maybe down to the fact that the cell is dependent on reading a DNA code to tell it, as it were, to tell the cell the precise order in which to join its amino acids together, precise order to get a polypeptide. Now, if there was a mistake in the DNA in the cell, and that we refer to that as a mutation or a mistake in the DNA, then that mistake would translate into a wrong sequence of amino acids in the polypeptide. And then if you had a wrong sequence of amino acids in the polypeptide, you wouldn't get the correct polypeptide, and the protein produced then wouldn't be correct. So a mistake in the DNA code, and the DNA sequence, the sequence of bases in the DNA, would give you then an error or a mistake in the order of the amino acids in the polypeptide, and that in turn would produce often what we refer to as a faulty protein, or a protein that doesn't work. And that protein might be an enzyme that doesn't work, or it may be faulty hemoglobin or some other faulty molecule made by the cell. And we'll look at that in a wee bit more detail when we talk about, when I do a video on DNA. But mostly that will be done in the upper sixth at A2. So the sequence of amino acids in the polypeptide is very important. The type of protein, or the protein that you get produced, depends on the order of those amino acids in the polypeptide. So at this stage, we're really just talking about making a polypeptide. And I'm just going to, just for illustration here, I'm just going to illustrate a polypeptide. It's just a long chain. This is just a piece of wire, but it's just to illustrate that a polypeptide is just a long chain of amino acids. If you want to just have a look there on page 15, you'll see there that the hormone insulin is a protein, and it's made up of two polypeptides. And you can see the two polypeptides there. You can see the polypeptides in that picture, and you can sort of see that you can sort of see the sequence of the amino acids. The amino acids there, just on the bottom polypeptide, read PHE, that's phenylalanine, and VL and ASN, and GLN and HIS, and so on. And that's the sequence of amino acids in that polypeptide. And you can see those polypeptides in that picture are about 20 to 30 amino acids long. But as I say, they can be hundreds of amino acids long. So that's a polypeptide. And we're gradually, as we're going along here, we're gradually working towards telling you how a protein is made. So at this stage, we're really just talking about making a polypeptide. Now, once the cell has made its polypeptide, then the polypeptide can undergo what you might describe as folding or curling up or coiling up into unusual shapes. Once a polypeptide is formed, it then undergoes various twistings and foldings to create what's called its secondary and tertiary structure. And then eventually you can put several polypeptides together to create the finished protein. And that finished protein would have what's called quaternary structure. So far we've just described primary structure, and that's just the order of the amino acids in the polypeptide. Now I'm going to point out that once the polypeptide is made, it then tends to twist itself up into some sort of shape. And the shape I'm going to illustrate first of all here is something called an alpha helix shape. And what I'm doing is I'm just winding the polypeptide, as it were, around this pencil, just to show you that this is what happens to the polypeptide. Once the polypeptide has been made by the cell, polypeptide may wind itself up or roll itself up into this kind of shape, twisted into, looks like a spring shape. But it's referred to as an alpha helix shape. So that's an alpha helix shape, looks like a spring. And this is an example then of what's referred to as secondary structure. The secondary structure of a polypeptide. One of the possible secondary structures is something called an alpha helix shape. So the amino acids the polypeptide sort of twists itself into this shape. And what you need to know about this shape is that this shape is maintained or held in place by hydrogen bonds. It's held in place by hydrogen bonds. So just to say a wee bit about bonds then, the original amino acids in the polypeptide are held together by peptide bonds. That's the bond between the amino acids. But this shape, or this twisted shape, this alpha helix shape, is maintained by hydrogen bonds. Hydrogen bonds between the amino acids that keep this shape. And the best way maybe to think about this is these hydrogen bonds are quite fragile. It can be easily broken. For example, if you were to heat this polypeptide, then you could possibly break the hydrogen bonds and the polypeptide would undo itself. It would lose its alpha helix shape. So this coil shape then is referred to as an alpha helix shape, and it's maintained by hydrogen bonds. Now there's an alternative what's called alternative secondary structure shape as well as alpha helix. You may also have another shape known as beta pleated sheets. And you can have both in the one polypeptide. So at one end of the polypeptide you can have this alpha helix shape, and then down the other end possibly you could have this beta pleated sheet. Now the beta pleated sheet is more difficult maybe to illustrate, but I illustrated just like that. A beta pleated shape, sort of like little bumps, almost like sort of snake shape like that. And once again, it's just a way of folding up the polypeptide. And that shape is also maintained by hydrogen bonds. So when you're asked about what kind of bond maintains or creates the secondary structure, and this is the secondary structure of the polypeptide, you'd say it's hydrogen bonds. Hydrogen bonds create the secondary structure of the polypeptide. Okay, just maybe looking at this point, maybe best to have a look at the one or two questions here. Question 1 on the front page of the booklet. You can see what type of reaction. So you see there in that picture, question 1, you see 2 amino acids, and you see they have 2 different R groups, R1 and R2. And identify the type of reaction represented by A and B. Well, joining the 2 amino acids together, that's a condensation reaction, so that's A. And B then would be, and I forgot to say that you can break if you have 2 amino acids joined together, you can break them apart by a hydrolysis reaction. The bond, A part 2, the type of bond then is a peptide bond that holds them together. and the product shown for reaction A is a dipeptide. I won't maybe go into too much detail into the answers to these questions because you have the answers, and the easier questions I'm going to leave you just to look at the answers. So it's a dipeptide. Question 4, just quickly looking at, sorry, not question 4, question, let's see where we're at. Yeah, sorry, question 4. Nope, not question 4, question 5. A very similar question to question 1. Two amino acids, you can see them been joined together by a condensation reaction. So X is a condensation reaction, Y is a hydrolysis reaction. Essentially, that question 5 mirrors question 1. It's just question 5. I think is a much older question. Question 1 is a more recent question, but you can see the two questions mimic each other. And it's so important to do the questions on this topic and on all topics. There's no point in just understanding the knowledge and the theory. It's very important that you do the questions as well. So question 5, condensation, X is condensation, Y is a hydrolysis reaction, the bond is a peptide bond. The final product of reaction X is a dipeptide. And part B then, what's meant by the primary structure of a protein? It says there primary structure of a protein. I would think of it more in terms of primary structure of a polypeptide. Really, that's what it means. And that's just the precise sequence, or precise order of amino acids present in the polypeptide. And describe the possible location of a sulfur group in an amino acid. As we've said, the sulfur group may be present in the R group in the amino acid. Okay, just looking then other possible questions then on the work we've done so far. 8, question 8, the elements present, list the elements present in amino acids, carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. Type of bond that links the amino acid together is a peptide bond. Suggests a function of the residue group. R is short for residue group, and you could say it gives the amino acid its identity, or it's involved, may contain a sulfur atom, which is involved in bonding in between amino acids, so-called disulfide bonds, and we'll come back to that in a minute or two. Distinguish between the term polypeptide and protein. Well, so far we've just talked about a polypeptide. This is a very old question. I don't particularly like the question, but if you were asked to distinguish between a polypeptide and a protein, you would say a polypeptide is a chain of amino acids joined together. That's a polypeptide. It turns out we'll learn about a protein, what a protein is in a minute or two. We'll discover that it usually involves several polypeptides joined together. So polypeptide is just amino acids joined one to the other. In a moment or two, we'll discover a protein is when you take several polypeptides and put them together to get the completed protein. So that's question 8. Question 10. We'll come back to question 10 in a minute or two. And the most important question here is question 13, which is an essay question about proteins. And essentially, question 13 encapsulates all the information we know about proteins, and it's a very good, it gives you 15 marks there to discuss the structure of proteins. So that's an extremely good question, and I'll try to go through that question in a moment or two once we've talked through the rest of the structure of the proteins. So far we've established that a polypeptide is a chain of amino acids joined together by peptide bonds. We've also established that once the polypeptide is formed, then it may spiral itself into a so-called alpha helix shape or a beta plated shape. You'll not be asked to describe either of these two shapes. What you will need to know is that they are two possible examples of secondary structure of a polypeptide. And the other thing you need to know is these are maintained, this structure is maintained by hydrogen bonds. So far so good. So we've got our polypeptide made, and it's got its secondary structure. Let's say, here's the next thing we discover. We discover that once a polypeptide has got its secondary structure, what the polypeptide will often do then is it will often roll itself up, cut itself up into a sort of a round or ball shape. So it's still got its alpha helix or beta plated sheet structure in the polypeptide, but the whole polypeptide has sort of rolled itself into like a ball, like so. And we refer to that aspect of a polypeptide as its tertiary structure. So the tertiary structure refers to where the polypeptide folds itself up into a precise three-dimensional shape, like so. This shape is also referred to as spherical shape or globular structure. Sometimes it's referred to as globular structure, a globular structure of a polypeptide. And this time we have to understand that there are several different bonds that maintains the polypeptide in this shape. And the bonds are involved here, and usually you have to list them. So there's hydrogen bonds involved again. There's also what are called disulfide bonds and ionic bonds, and something called hydrophobic interactions. You're not usually asked to describe any of these bonds. You simply have to know that there are. An exam question typically they want to know about three of them. So you'd say hydrogen bonds, disulfide bonds, and ionic bonds. The disulfide bonds, if you look there on page 15, you can see the disulfide bonds are usually formed between the sulfurs in the R groups of adjacent amino acids. And you'll see the amino acids there on page 15 at the top of the page. You'll see that the amino acids that have a sulfur are CYS, that's cysteine, C-Y-S-T-E-I-N-E. I think cysteine is the only example there in that picture. So cysteine has a sulfur group, and that sulfur group can join to another, make a bond with another sulfur group in another amino acid. And that maintains involved in maintaining the tertiary structure of the polypeptide. So this tertiary structure is held in place by these bonds. Now, the understanding these bonds is important, and the best way to understand them is that they're not very strong, any of these bonds, and that if you were to heat this polypeptide, then what would happen is the bonds would break and the polypeptide would unravel from its shape. So it would start to lose its shape. Your polypeptide would start to lose its shape. And if it loses its shape, then that usually means that the polypeptide is no longer functional. Because what we'll discover is proteins and polypeptides and these kind of molecules are all about shape. So often proteins are about shape. If you think of the important proteins that you know, well, the obvious one is an enzyme. Enzymes are all about shape. Enzyme has to be the precise shape so that its active site is the right shape, so that it's complementary to its substrate molecule. If you change the shape of the protein, in this case the polypeptide is rolled up, then you change the shape of its active site and no longer functions. Hemoglobin, another important protein, it's all about shape too. Hemoglobin binds to molecules of oxygen. If you change the shape of the hemoglobin molecule, even ever so slightly, then the hemoglobin molecule doesn't bind correctly to oxygen and you don't get correct oxygen transport around the body, and so on and so forth. All proteins, nearly wherever you come across them, it's their shape that's important, and anything that modifies their shape or changes their shape or makes their shape wrong makes the protein faulty or ineffective. Now, we've come to a very important point. At this stage, so far we've just been talking about, we've talked then about primary, secondary and tertiary structure. And those three words here relate to the structure of a polypeptide. So there's a polypeptide, and if you could see inside it, well, first of all, it's got primary structure because you could, if you unravel it, you'll see it's got a precise sequence of amino acids. It's got secondary structure. It may not be so visible now, but you can still see the alpha helix shape there of the polypeptide, so that indicates it's got secondary structure. And the fact that it's rolled up into a ball shape, that gives it its tertiary structure, its globular tertiary structure. But notice so far that we've only been talking about one polypeptide, and I've been trying to steer away from the word protein at this stage. Well, now we come to something very significant. What I'm going to say next is that most polypeptides don't tend to exist on their own as a single polypeptide. Usually what you find is, here I've got another polypeptide, so there's the original one. I'm making another polypeptide with, it's got an alpha helix secondary structure there, and I'm going to roll it up into a ball so that it's got tertiary structure. And what I'm going to say is that most polypeptides don't exist on their own, but they exist along with another polypeptide, at least one more polypeptide. In fact, it can be several polypeptides together. So I've just, from my pocket here, I've just produced another polypeptide. So here, for example, is 3 polypeptides, and they're all attached together. These 3 polypeptides are attached. It's a bit of a mess at this stage, but these 3 polypeptides are attached together, and with 3 polypeptides, we've now got the protein, because typically proteins are constructed of several polypeptides held together to create the protein. And when you have several polypeptides together, we refer to this as the quaternary structure. The quaternary structure. We would say if a protein has more than one polypeptide, we say it has quaternary structure. In some cases, the protein may just be one polypeptide. So there's the original polypeptide. An example might be an enzyme. An enzyme may consist of just one single polypeptide. So there's an enzyme there, single polypeptide. There might be the enzyme's active site, maybe just some hole on the surface of the polypeptide. So this could, if I said this is an enzyme, it's a protein, it consists of just one polypeptide. But here I've got another protein, let's say a different enzyme or a different protein, and it's made up of 3 polypeptides stuck together, as it were. So if a protein just consists of one polypeptide, then you can only say that that protein or that polypeptide has primary structure, secondary structure, and tertiary structure, but it doesn't have quaternary structure. If there's just one polypeptide involved, it doesn't have what's called quaternary structure. For a protein to have quaternary structure, it must involve several polypeptides held together or stuck together, and then it has quaternary structure. And you'll notice there at the bottom of page 14, I say most proteins consist of 2 or more polypeptides bonded together, held together. And in this case, then the protein is said to have quaternary structure. And you can see a protein there that looks like it's got 3 polypeptides stuck together. 3 polypeptides gives it quaternary structure. Now, at this stage, we still don't, in many cases, have the completed protein. Because what you'll find is that many proteins, as well as having, let's say, 3 polypeptides or 2 polypeptides or 4 polypeptides, they'll also have what you refer to as a non-protein grouping or groupings attached, and these are referred to as prosthetic groups. So this is like an extra additional molecule that needs to be attached to make the protein, as it were, complete or functional. And this extra, it's a non-protein group, so it's not made of amino acids, it's made of something else, made of some other molecule, and it's referred to as a prosthetic group. And once you have, when a protein does have these prosthetic groups, it's referred to as a conjugated protein. So there's a lot of words here. Prosthetic group is the extra group that's attached to the protein. And whenever the protein does have a prosthetic group, it's referred to as a conjugated protein. Now, a classic example of a conjugated protein is a conjugated protein with quaternary structure is hemoglobin. Hemoglobin is made up of 1, 2, 3, 4 polypeptides. So 4 polypeptides attached to each other, 4 polypeptides. So the fact each polypeptide has primary, secondary, and tertiary structure. And the completed protein then is made up of 4 polypeptides, so it's said to have quaternary structure, and it's said to be a quaternary protein. As well as that, then attached to complete the hemoglobin, you have these non-protein, what are called heme groups, and you have a heme group attached to each polypeptide. And the heme groups then are prosthetic groups, and they create then what makes the hemoglobin what you call a conjugated protein. So conjugated protein is only called that if the protein contains these non-protein parts called heme groups. Now, there are only 2 or 3 examples of of prosthetic groups that you would have attached to proteins. So hemoglobin is the classic example, in which case the prosthetic groups are the heme groups. And these heme groups, by the way, contain an atom of iron. This is why iron is often considered to be important for blood production, because iron is part of the heme group, which is part of the hemoglobin molecule, which is part of a red blood cell, and therefore iron is needed in your diet to provide for these heme groups, to provide you with sufficient hemoglobin for your red blood cells. So that's the basic structure then. So polypeptides with a primary, secondary and tertiary structure, and then if you've got several polypeptides attached together to create a protein, you see that protein's got quaternary structure. If the protein consists of just one polypeptide, then it can't have quaternary structure. Okay, now I'm going to specifically refer then to mostly to question 10. I'm just going to quick look at question 10 there. Question 10 is quite an old question. It talked about 2 proteins that we know about. Maybe don't know about myoglobin at this stage, but we know about hemoglobin. And it tells you about hemoglobin, and then it tells you about another protein called myoglobin. Myoglobin is very similar to hemoglobin, except myoglobin doesn't have 4 polypeptides. Myoglobin just has 1 polypeptide. It just consists of 1 polypeptide, but it also has a heme group, and it's related to hemoglobin, and you'll come across it in another topic. And it asks you then to discuss hemoglobin and myoglobin as to whether they have secondary structure, tertiary structure, quaternary structure, etc. And you'll see that secondary structure. So if you read about both molecules there, you'll discover that both molecules, it says both molecules have what are called alpha helixes. And alpha helix is the spiral-shaped structure that you get in the polypeptide. So alpha helix structure. So you can tick both of them have that. So you can tick that. It has a quaternary structure. Well, as I said, you can only have quaternary structure if you have more than 1 polypeptide. So if you say you had 2 or 3 or 4, myoglobin doesn't. Myoglobin just tells you there myoglobin. It consists of a heme group, but it only has 1 polypeptide. So you see it doesn't have quaternary structure. So you can tick yes for hemoglobin. Is a conjugated protein? Well, a conjugated protein is where you have the presence of a non-protein molecule. In this case, both of these have heme groups. You can tick the same for both of them. Types of bonds that contribute to the tertiary structure of a protein. This is question 10, by the way. That would be, you could say, hydrogen bonds, ionic bonds, or disulfide bonds. Okay, so we'll leave that at this stage. I'm looking then specifically now at question 13. Question 13 is a very recent question. I think it's 2019, module 1 essay question, or long question in the essay paper. Basically, it's a very good essay question, a very good question that covers all of this topic. Now, I would specifically ask you to have open there page 16 of my notes there, where I'm looking at comparing a couple of very important proteins, collagen and enzymes. And that's essentially what's on that page 14 there, page 16. That covers all you need to know essentially for this essay, to actually get all the points in this essay. And it's useful then to have if you have the answers for the essay there. You can open the mark scheme, and you can see how many points there are there. And I'm just looking just to see there are in total 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, about 19 points given in the mark scheme to answer that essay, and you needed 15 points. So it's quite tight. You need to know your stuff essentially. And it says, the essay says, describe and explain how a wide variety of structures is achieved in proteins. And then it wants you to go on and talk about collagen, enzymes and hemoglobin. So but the first part is talking about the structures present in proteins. So that's the primary, secondary, tertiary, quaternary structure in the making of a protein, or in the construction of a protein. And I see there just in total they give you 1, 2, 3, 4, 5, 6, 7, 8, 9 marks for the structure. of the protein, which is quite okay. So you need 9 points to do with structure of protein. And so if you follow the mark scheme down there, it says, we'll just run through the points quite quickly. The primary structure consists of a sequence of amino acids. And so the primary structure refers to the sequence of amino acids in the polypeptide. And mentions there the polypeptide. The amino acids are joined with peptide bonds. And then it says something very, very important in the second point. It says the type of protein formed, or the protein that is formed, depends on that sequence of amino acids in the polypeptide. So if you change the sequence of amino acids, you get a different protein, you get a different type of protein, or you get a faulty protein, or a protein that doesn't work. The type of protein then depends on the order of the amino acids. The secondary structure then it can be alpha helix or beta plated sheets. So you just have to know those two structures. You have to be able to say alpha helix or beta plated sheets. And you would say that those structures are maintained, the polypeptide is maintained in those structures by hydrogen bonds. And that's, I think that's point, that's the third point. And point number 5 there, bonds twist the amino acid chains into a helical or spiral shape. Now, so point 5 there is just giving you a chance to say what the alpha helix, for example, is. That the alpha helix is the polypeptide is folded into a helical shape, and it's the making of the hydrogen bonds. As the hydrogen bonds make, they pull the polypeptide into that shape, into that helical shape. Point 6 then goes on to talk about the tertiary structure. It's basically saying then that the polypeptide can fold itself into, it says tertiary structure gives the protein its three-dimensional shape. In other words, it's gone from being a long, slender polypeptide coiled up, let's say, in an alpha helix shape, it's now in a sort of roundy, three-dimensional ball shape or spherical shape or globular. Globular is a good word, globular shape. And then you get a mark for saying the kinds of bonds that are involved in keeping the protein in this three-dimensional shape, this globular shape. Hydrogen bonds, ionic bonds, disulfide bridges, you could say disulfide bonds, and hydrophobic interactions. Don't worry about the hydrophobic interactions, how they work. You must mention these 3, so you have to quote 3 of those, so I quote the first 3. Then you move on to the next bit, which is the quaternary structure, and you would simply say then that several polypeptides come together to form the completed protein. And when you have several polypeptides together, you say that the protein has quaternary structure. What way they said? They've just said a quaternary structure occurs or is present if 2 or more polypeptides are involved. So you've got more than one polypeptide involved in the structure, then you've got a so-called quaternary protein. And then it says some proteins also contain prosthetic groups. In such case, they're called conjugated proteins. So you mention both those words. So then that's quite a nice little summary of the protein structures. It then asks you to, it says, to discuss the structure of collagen enzymes and hemoglobin and relate their structure to their function. And they're giving you, so you're getting in, I notice there in total about 10 marks for those 3. So effectively you should be thinking, I need to come up with 3 points about each of those. Collagen enzymes, and hemoglobin. Now, on my table on page, I think it's page 16 again, you'll see there I've discussed collagen. I've said it's a long fiber-like protein found in tendons and skin. It's an example of what's called a fibrous protein. So a fibrous protein is a protein that isn't rolled up into a ball shape, but it may consist of polypeptides that are wrapped around each other to create like a long, slender protein. So, and collagen is such a long, slender protein. It actually consists of 3 polypeptides coiled around each other, and the polypeptides are held to each other by hydrogen bonds. Each polypeptide has secondary structure of alpha helixes. There's no tertiary structure. The polypeptides are not rolled into any kind of ball or three-dimensional shape. They're simply long, no tertiary structure, but you do have quaternary structure in the completed protein because this involves more than one polypeptide. So just notice that the protein collagen, the completed protein, has polypeptides with primary and secondary structure, but the polypeptides don't have any tertiary structure. But then you've got quaternary structure because you've got more than one polypeptide involved. And you have to relate this then to the function. So I'm just reading about collagen. It says the mark scheme says collagen is a fibrous protein consisting of 3 polypeptide chains, and the chains are cross-linked. That simply means that, you see in my notes I've said they've got hydrogen bonds between the chains. That's this cross-link. So if you want to write in cross-links above hydrogen bonds, and that gives the protein its high tensile strength. High tensile strength means the protein is stretchy. It's resistant to stretching. It's got good strength, and that's because of the three polypeptides wrapped around each other. Each polypeptide on its own wouldn't be strong enough. But if you've got 3 wrapped around each other and they're held together by hydrogen bonds, that gives them a lot of strength then. So that's the marks for collagen. Enzymes. Typically enzymes may just consist of one polypeptide. So there may just be one polypeptide rolled into a globular shape. And you see in my notes there I've got a picture of an enzyme. It said this enzyme is a single polypeptide folded into a spherical 3D shape. The polypeptide has primary, secondary and tertiary structure. But it has no quaternary structure, this particular polypeptide, because there's only one, this particular protein, because the protein consists of just one polypeptide, no quaternary structure. So it says enzymes and hemoglobin are both globular proteins. Hemoglobin is also a globular protein because its polypeptides are folded into this 3-dimensional shape. And then says the variety of ways in which the polypeptide can fold gives rise to a variety of shapes. So different 3-dimensional shapes of the polypeptide gives different shapes of the enzyme. And then you have to say something about the shape of the enzyme determines the shape of its active site. And then you can say that the active site has a precise shape into which a substrate molecule might fit. And then, yeah, that gets 2 marks. So the shape of the active site confers specificity. And you would say something like each polypeptide has a precise shape into which only one substrate molecule can fit. And you can refer to the lock and key hypothesis or the just fit hypothesis. And then that's an enzyme, 3 marks. And hemoglobin is the last item there discussed in the mark scheme. It says hemoglobin consists of 1, 2, 3, 4 polypeptides. Each polypeptide contains a heme group. And by the way, that hemoglobin molecule can carry 4 molecules of oxygen. Each hemoglobin molecule can carry 4 oxygen molecules. Yes, a heme group allows hemoglobin to transport oxygen. So you get an oxygen molecule attaching to each heme group. So each hemoglobin molecule can carry 4 molecules of oxygen. So those are 3 important proteins, 3 types of proteins. Then the fibrous protein, which would be collagen, and the other 2 are referred to as globular proteins. But enzyme is a good example of a globular protein, and then hemoglobin is a very important protein. Other examples of, just maybe before we finish, other examples of conjugated proteins. So I said a conjugated protein is a protein where you have non-protein molecules attached. If, for example, hemoglobin has heme groups attached. Other examples of conjugated proteins, really the only other examples we come across would be in the cell membrane, where you have phospholipids in the cell membrane, and then you also in the cell membrane you have proteins. So say that's the cell membrane there. There's some phospholipids and there's a protein. Let's say that's the cell. And these proteins will often have, proteins in the cell membrane will often be so-called conjugated proteins because they'll often have a non-protein part attached to the outside of the protein. And that non-protein part might be a chain of glucose molecules. And that chain of glucose molecules, that's the prosthetic group essentially. So this is the protein part, and then this is the non-protein part. And this part on the outside here is often referred to as glyco. So that's the prosthetic glyco, and that makes this a glycoprotein. So a glycoprotein is a protein where the prosthetic group is made up of some carbohydrate molecules, for example, glucoses. So you refer to as a glycoprotein. Equally, this non-protein group might be made up of lipid molecules, and that gives you then a lipoprotein. So glycoproteins and lipoproteins are both examples of conjugated proteins, where the non-protein the non-protein prosthetic grip may be made up of carbohydrates that gives you a glycoprotein, or lipids gives you a lipoprotein. And so that's the structure of proteins. Maybe the only other molecule to look at here is DNA, nucleic acids, and I'll look at that in a future video. Yeah, just one other thing maybe to look at here, and just briefly, and that is prions. So if you turn to page 18 in the notes here, prions, and the only question relates to that would be question 1. Question 1, the second part of question 1 there, so if you open that up, that's, we'll have a look at that second half of question 1. And prions are abnormal proteins that, I like the idea of an abnormal protein is a protein that hasn't got the correct shape and is not functioning properly. Abnormal proteins, these proteins, when they get into the human body, they cause a disease. And the disease in humans is called Creutzfeldt-Jakob's disease, CJD. It occurs also in other animals, where it's called by different names. In sheep, I think it's called scrapie, and in cows, it's called bovine spongiform encephalitis, or BSE. It used to be referred to as mad cow disease. And this is a disease that occurs sometimes spontaneously in humans, but it's also associated with eating meat from animals with the disease. Now, these abnormal proteins are called prions, P-R-I-O-N-S, disease, abnormal proteins that cause disease in the human body. And the part of the human body that they affect is the brain, nervous tissue. And the first question you might ask is, what's wrong with these proteins that makes them abnormal? Well, at the bottom of page 18 there, bottom of page 18, you see a picture of a normal protein and a prion protein. And here's what's unusual with prion proteins. Normal proteins will have, in their secondary structure, their polypeptides will have both alpha helixes and beta plated sheets. The prion protein, however, will have an excess of beta plated sheets. So it'll be a protein where its secondary structure has been modified. So instead of having lots of alpha helixes in the structure, it's got lots of beta plated sheet structure. So its structure with its polypeptide is changed. And that makes the protein non-functional and disease producing. Just looking at the questions, dip the specific type of molecule that causes this disease, and the answer is a specific molecule of prion, and it's a type of protein, but it's an abnormal protein. And you have to name other diseases there. So BSE and scrapie are two diseases. And one way in which you could become infected with such proteins, well, eat meat from animals that have had the disease, and then once the proteins get into your body, they don't tend to cause an immediate disease. They may have a long, what's called incubation period, and that simply means that the proteins may be present in your body for years, maybe upwards of 20 years, before you start to actually exhibit symptoms. What do these proteins do when they get into your body? Well, it turns out that they sort of infect your own proteins, and they cause your own proteins to become prions. And the proteins that they attack specifically are proteins in the cell membranes of nerve cells. And they cause these proteins to become prions, and that these proteins are no longer functional. So this is a disease of the nervous system. State what happens during the incubation period. During the incubation period, you get your normal proteins in your body converted into prions by the prions that you've taken in your food. So normal proteins, during the incubation period, your normal proteins lose their alpha helix shapes and gain lots of beta plated sheets instead. And the normal proteins your normal proteins effectively become prions. So you may get a question from time to time about prions. But that question once is the only question that has ever appeared. Okay, there's some practical aspects to do with proteins, but I'll leave them until a later time. As I say, I've done another video which is looked at carbohydrates and lipids, and that's available as well to anybody who's doing tertiary. So there you are. I'll leave the practical work on proteins until there. Thank you very much then. Bye.